today. So for today's video, I am going to tackle about the protein synthesis, which is all about transcription. So keep on watching. The first step in protein synthesis is the transcription of mRNA from DNA gene in the nucleus. At some prior time, the various other types of RNA have been synthesized using the appropriate DNA. The RNAs migrate from the nucleus into the cytoplasm. So, what is a transcription? Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA or mRNA. Transcription also is the first of several steps of DNA-based gene expression in which a particular segment of DNA is copied into RNA, especially mRNA, by the enzyme RNA polymerase. During transcription, the DNA can be safely and stably stored genetic material in the nuclei of cells as a reference or template. Meanwhile, mRNA is comparable to a copy from a reference book because it carries the same information as DNA, but it is not used for long-term storage and can freely exit the nucleus. So what is the main goal of transcription? So, the main goal of transcription is to make a RNA copy of a gene DNA sequence. For a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide or protein subunit. Eukaryotic transcripts need to go through some processing steps before translation into proteins. Let's cover the process of transcription. So, Kornberg and his research group have made several fundamental discoveries concerning the mechanism and regulation of eukaryotic transcription. Transcription is carried out by an enzyme called RNA polymerase and a number of accessory proteins called transcription factors. Transcription factors can bind to a specific DNA sequence called enhancer and promoter sequence in order to recruit RNA polymerase to an appropriate transcription site. RNA polymerase synthesizes RNA by following a strand of DNA. RNA polymerase is an enzyme that is responsible for copying a DNA sequence into RNA sequence during the process of transcription. Transcription involves 4 steps: initiation, elongation, termination, and processing. Initiation is the DNA molecule unwinds and separates to form a small open complex. RNA polymerase binds to the promoter of the template. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so that enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. So, transcription elongation is a regulated process in which an RNA chain complementary to the template strand of DNA is synthesized as RNA polymerase moves along DNA. Typically, elongation is the second process of transcription. Elongation. RNA polymerase moves along the template strand synthesizing an mRNA molecule. In prokaryotes, RNA polymerase is a hollow enzyme consisting of a number of subunits, including a sigma factor that recognizes the promoter. In eukaryotes, there are 3 RNA polymerases, 1, 2, and 3. The process includes a proofreading mechanism. Termination is the ending of transcription and occurs when RNA polymerase crosses a stop sequence in the gene. The mRNA strand is complete and it detaches from DNA. The last steps of transcription is the processing. So after transcription, the RNA molecule is processed in a number of ways. The introns are removed and the exons are spliced together to form a mature mRNA molecule consisting of a single protein coding sequence. RNA synthesis involved the normal base pairing rules, but the base thymine is replaced with the base uracil. Translation is very important in the process of making proteins. Without transcription and translation, your body would have no possible way to make proteins or function. Proteins allow your body to do everything. Trivia. What is the end result of transcription? So, the DNA is converted to the strand of mRNA to be translated in the ribosome.
It's caught in the crossfire. It's caught in the crossfire.